Hi, everybody. Welcome to our webinar this week. I'm Shravan. I'm currently working as Health Engagement Officer with Macular Disease Foundation Australia. Thank you for joining us today. We are very fortunate to be joined by Professor Pébay, who will be taking us through a research update with Macular Disease Foundation Australia grant recipient. Before we move on, I would like to acknowledge the traditional custodians of the land on which we are meeting today and pay my respects to elders past, present, and emerging. So I would like to start with some housekeeping for the webinar. So if you do have any audio or visual problems, please refresh your page, or you can always call us on 1-800-111-709. If you're hearing me, this means you have been able to access the webinar. Please, because we have a number of people attending, everyone's microphone has been muted. So we won't be able to hear you. Videos has also been switched off, so we can't see you. To interact with the group, please ask questions on Q&A box, which you'll find on the right side of the screen. So I'm showing a screenshot of where to find those features. At the bottom of your screen, you'll be able to see those icons like chat, raise hand, and Q&A. Today, you'll be using this Q&A functions mostly. Your questions will be answered at the end of the presentations, but feel free to type your questions at any time. I should note that other attendees will not be able to see your questions. Only myself and Alice will be able to see those questions. There is also an option to send your questions anonymously as shown here. And if everybody could keep the questions general in nature, as we can't provide personal advice over the webinar. So if you're having any audio or video issues, please call on our toll-free helpline on 1-800-111-709 instead. And finally, there is a last icon, leave meeting. So clicking on this one will allow you to exit the webinar. And again, welcome everyone. Thanks for joining this webinar. I would like to take the opportunity to introduce Professor Alice. Professor Alice Pébay obtained a PhD in neuroscience from the University of Paris in 2001, and subsequently joined Monash University to undertake research on human pluripotent stem cells. She then continued her research in the area at the University of Melbourne, where she commenced in 2007. Alice team aims to use patient-specific pluripotent stem cells to model neurodegenerative disease, in particular age-related macular degeneration. Alice was awarded a National Health and Medical Research Council, NHMRC, Career Development Fellowship in 2012. Subsequently, an Australian Research Council Future Fellowship in 2014, and is now an NHMRC Senior Fellowship. Alice is the primary inventor of 3 granted international patents related to stem cell technology. Now, let me welcome Dr. Alice Pébay. Thanks, Alice.
Thank you very much, and good afternoon, everyone on the other side of the screen. It's always a real pleasure to be able to work with the Macular Disease Foundation Australia. And as a researcher, I've really enjoyed my time working with the foundation. So what I would like today is to talk to you about the work we do in the lab, which is really about fighting blindness. We are doing our best to use what we call stem cells to fight blindness. And in particular, we are trying to understand how blindness happens using cells in a plastic dish. I'm going to use the pointer. I hope you can see the laser pointer here. It is okay. So before I go on about the work we do, there's just a few things I would like to remind people so that we're all on the same page when we talk about a cell, because we talk about stem cell, but first of all, what is a cell? So if we look at our body, basically, we're all made of exactly the same stuff. We are made of cells, of billions of cells that have different functions and different shape and personalities, I would say, depending on what they have to accomplish in our body. When the cells start to pack up together, they start forming tissue, such as muscle tissue, and then they pack even more and then become organs organs and eventually contribute to a system that we have in us. It's the same for the eye. The eye is an organ that is composed of different tissues, which are themselves composed of cells that make the eye cells. Now, we were not made that way. Development happens with fecundation, and we don't start with all those organs and all those cells ready to go. No, it doesn't happen like that. What happens is that at the start of life, as a human, what happens is that there is one cell that is called a stem cell that proliferate, make a lot of stem cells, but also make some daughter cells that differentiate and can become those different cell types that will eventually form the different organs of the body. So these cells, these stem cells, are really fundamental to who we are, because without them, there wouldn't be all these structures that make us, all these hierarchies that we see in us. These stem cells, a different type of stem cells, some that are found in us very early in development, which we call pluripotent stem cells. And we also have, as adults, and as we age in life, we still have pools of stem cell in our body, which we call adult stem cell, that help some part of our body regenerate. You would have heard that you've got some blood stem cells, for instance. So there are some stem cells left in us in our body, but they don't all have the same capacity. But just to go back to a stem cell, because we'll talk about it, it is a cell that in effect has not become anything and has the potential to give rise to everything. So we can understand why these cells are so important in medicine, because if we know how to use those cells, if we know how to harness their power, we might be able to ask a stem cell to become an eye cell that has degenerated in a disease, and then replace that eye cell with the one we've made in a dish as a cell replacement therapy. So these are very powerful cells to work with. Another aspect I would like to talk about before we go further is when we look even more down onto a cell, more we focus like with a microscope onto a cell, all cells are basically made of the same stuff, even if they have different function. There's two little aspects in a cell that I would like to talk about. One of them is that every cell in our body contains all the information that makes us, that is called genetic information. This genetic information is found in a part of the cell called the nucleus, which has the DNA, which is basically a very long thread of information that is compacted into that nucleus in the shape of genes. Every cell has all this information within the cell. Now, we don't need all the information all the time. A muscle cell will only need the information that relates to a muscle cell, life and function, and an eye cell will also only need the information that is necessary to be an eye cell. But all cells, despite only needing some functions, have all the dictionary basically in them, and that's important for later work. I will show you. The other aspect I want to mention about a cell is that, like a car, a cell cannot work without energy. And energy is made in every single cell of ours in a little factory, which we call a mitochondria. The mitochondria is the factory of the cell. And without that little organelle, the cells cannot work because there's no energy to sustain the life of the cell. Again, if the mitochondria doesn't work well, there's less energy available for the cells and the cells suffer a little bit. Again, we'll talk about that later. So those were the concepts I wanted to bring before we go further. The eye, as I've said just before, is an organ and it's made of cells because we are made of cells. But the eye is quite an interesting structure, really. And for most of us, the eye is that structure that allows us to make visual sense of the world around us. And when we think about how this works, it's quite incredible because we have a wavelength. So this is just a wave of light that is coming into the eye and eventually tap the back of the eye, which is called the retina, the neural retina. In the neural retina, if we look into a microscope, what we would see is that it's not a simple structure. It's made of multiple different types of cells, which all play a very important role in getting that light signal transformed into an electrical signal that can then travel back to the back of the brain through the optic nerve. And at the back of the brain, it will start the integrating process by the brain that will try to understand the signals that are around us. Now, in various eye conditions, retina and retinal cells are affected. And in age-related macular degeneration, the two main cell types that are affected are called the retinal pigment epithelium, which is that back structure at the back of the eye, the brown structure we even see, or the RPE. We don't know how and why, but these cells here, these very thin cells, die in AMD. And we need to understand why, because when they die, they take with them eventually those very sensitive cells on top, which are called the photoreceptor. And the photoreceptor are basically receptors for photons, so for light. These are the cells that allow us to see. They're the first one to transform light into a signal the brain recognizes. So when these cells go, or when they're not happy, we don't see as well. And this is what happened in AMD. Now, going back to stem cells, why do you think these cells are important? Well, because I said before, stem cells can become everything we want. So we have the potential, if we know how to make them properly and guide them properly, we can ask those cells to become the cell of the eye that die in a disease. And we can also ask cells to do more important things, which I will talk about in a minute. But before we needed to do that, we needed to have a good source of cells, of stem cells that would be able to use in the laboratory. And this person here is Professor Yamanaka, who in 2007 demonstrated something really remarkable. He showed with his team that you could take any cell of our body, so basically a piece of skin or a bit of blood, and ask these cells, through addition of some special food onto them, to go back in time into a stem cell. That is what we call reprogramming. And that was really amazing because thanks to Professor Yamanaka, we can make stem cells from every single one of us who wants to have stem cells for research, for instance. But for the big picture, everybody could have access to their own stem cells from now on. And having access to their own stem cells means that we could potentially be able to make cells that are affected in whichever condition which will affect us, including for the eye. If we make those cells, we can imagine that those cells could then be reimplanted in organs that are affected in diseases. Here is an example of stem cells we've made in the lab from people's skin, and that we differentiated eventually into the retinal cells that are affected in AMD, which are called the RPE cells. They're very, very dark, as you would see in people's eyes. It's the dark cells we get in the eye. Now, these cells, not our cells, but around the world, these type of cells have already been used in clinical trial for age-related macular degeneration. What people did is that they took stem cells from patients, made them, guided the cells into making these retinal cells that degenerate in AMD, and then implanted them back in people to see whether or not it was a safe way to replace the cells that had died and if it was effective. This is happening in America, this is happening in Europe, this is happening in Japan. The clinical trials have started. So this is very, very encouraging aspect of stem cell research. Now what we do in the lab is a little bit different. We use stem cells not to replace, but to try to understand what goes wrong in AMD. In fact, we don't know much about AMD still in a dish, because in AMD, in full stop. The reason we don't really understand AMD today is because we don't have great models to study AMD in a laboratory. We can use some models at times, but they're not really good. And one of the limitations, of course, is we can't go and take the back of the eye of someone to study what goes wrong. It's unethical. We cannot do this. But thanks to stem cells, together with my colleague Alex Hewitt, who is an ophthalmologist, we thought, hang on, what about we take people's stem cells, we make them from their skin, we take patients' skin, make them into stem cells, then ask those stem cells to become the cell affected in AMD, and then look at what is different between healthy individuals and people with AMD. Now, AMD is a complex condition because AMD is not due to just one gene or one factor. It's multifactorial. We know that some genetic factors, some of those genes that we have in all of our cells, are involved in AMD. They are risk factors to AMD. We also know that the environment is probably a risk factor. Aging is a risk factor. The diet, smoking, all of these are complex factors that might push someone towards the AMD, bring AMD to someone. So because it is a complex condition, we cannot study this condition with 1 or 2 or 3 samples. We knew we had to take a large amount of samples to study AMD. And with Alex Hewitt, when we started this work over 10 years ago, it was really risky because nobody had done it. People had studied a more simple condition, which we call monogenic condition, where it's just one gene that's involved, or developmental condition, but not something as complex as AMD in a dish. And with Alex, we knew that to do this type of work, we needed to change the way we were working, because having hundreds of patients, stem cells in a lab meant we couldn't work with people in each hood doing their own little work. We needed something much more efficient. We needed a robot. The robot didn't exist 10 years ago. And with Alex, with eventually sufficient work done, we were able to obtain funds to make a robot with a company called TCAN. We made a really good robot that was able to maintain all our cells, all our stem cells that we wanted to do, which was much more efficient than if humans were to do it. Just a little joke, we are based in Melbourne. We thought if we don't get enough money at the end of our work to continue the work, at least we will use the robot to make coffee and people will be happy. But we didn't have to go to that extent. In fact, we had sufficient funding and sufficient work to do. We were able to use this robot, which was new in Australia, especially, to go on with the research we wanted to do, the important research we wanted to do. And in fact, here is the team of all the people who contributed to this robot working to get our hundreds of patient stem cells made on the robot in a way that was reliable. In parallel, so we patented this work, in fact. It is now being reproduced in Denmark to do very similar work to the work we did in Australia years ago. And thanks to this robot, we generated over 600 patient stem cell lines, most of them with ophthalmic disease, including, thanks to the MDFA, over 60 lines with geographic atrophy, which is a late-stage AMD phenotype. In parallel to making the stem cell, we needed also to work to make sure that we could ask the stem cell to efficiently become the cells that are affected in AMD, which are these RPE cells. And we have really good protocol now that we made in the lab to get those really mature, really healthy RPE cells in the lab so we could start our work on AMD. And really things started changing in 2017 when my team received the MDFA research prize. It changed what we could do because we finally were financially able to generate that large bank. We had set up the robot, we knew we could do the work, we knew we could make the cells, we knew we could record what we wanted on the cell, but we needed the funds to be able to make it work on the large scale. And that was given to us, this opportunity, thanks to the MDFA. And what we did with the funding from the MDFA was to generate stem cells from roughly 120 individuals. But at the end, when with all the quality control, we had a large number of healthy individuals and a large number of AMD individuals. We did the stem cells, we made the retinal cells, and we started comparing those samples. What was different in the geographic atrophy, so in the AMD samples compared to control. And to make it short, basically, we were happy, of course, because it meant the work worked and the bet was right, but we could see clear differences between the control patients and the AMD patient cells. And in particular, what we could see was that one of the things that was really affected in the AMD cells was the mitochondria. That little factory, energy factory of the cells. In every assay we tried, we looked at the genetic level, so at the gene, at the protein level, at the functional level, at every level we tried, we saw that the mitochondria is working very differently in the samples that came from people with geographic atrophy. So this is really for us a good demonstration that there's something now that we've identified in the mitochondria that is different. Perhaps the cells from the start don't breathe as well, and so with time they breathe less and less, and it's much harder to do their job, and maybe that's why cells start dying in AMD. And what do we do with this now that we've discovered this data? Well, we can now have a model in which we can study how to revert that phenotype. And that's what we are trying to do. Again, this time with MDFA, not as a funder, but as a partner. We went to the Medical Research Future Fund and got funding to take this discovery and start screening on drugs that are able to help us identify a way for the cells to do better. So that's one of the work we are currently doing in the lab. So using our data, our results to try to find potential treatments. Another thing we're trying to do in the lab, and you would have heard about artificial intelligence, is to try to find an unbiased way where we can identify what goes wrong in the cells that our eyes can't see properly, or our brain can't comprehend itself. So we're using AI to help us identify subtle differences between the AMD samples and the control samples. So we're doing a lot of stuff now, which is really not at the stage of looking at what went wrong, but on how we can fix what is wrong in the cells. And this is really the interest of my lab, trying to understand how conditions happen with the hope that we can then find treatment perhaps to prevent those conditions from happening down the track. And with this, I would like to thank every single one of my colleagues who have worked over the years in different institutions on this project. I would like, of course, to thank the funding that supported this work, and in particular the MDFA, because without the MDFA, this work would just not have happened. And most importantly, I want to thank all the patients who gave us the skin biopsies, as well as a bit of blood for us to do our work. It was, and I don't know if in the room there are anyone who gave us the skin, but if they are, well, please know that your skin has helped us make some really important discoveries that I'm very happy to report to you today. Thank you very much.
Thanks, Alice. Thank you so much for this. It was extremely informative, and I think we all learned a lot. So just a reminder that if you have any questions about anything Alice went through, please click on the Q&A icon there and type your questions into the white box and click send. I'll be reading out the questions very shortly. As I mentioned before, all the other attendees will not be able to see your questions, only myself and Alice. And please try to keep the questions general in nature. So now, before I move on to the questions, I would like to talk about the work of Macular Disease Foundation Australia. The main goal of our Macular Disease Foundation is to reduce the incidence and impact of macular disease. And we do achieve that through a number of pillars that guide our work. So this includes prevention and early detections. So in this, we provide accurate, current, and ongoing information to increase the awareness of macular disease nationwide. Just last year, we reached almost 7,000 people through education sessions. So the next one is support and services. In this pillar, we provide support for all our patients and also facilitate access to other support services, including the support groups, our helpline, and community educations, among other things. And we are also the voice of macular disease community. It's our privilege to advocate for the best interest for the macular community, whether it be designing policies for the state and federal governments or making sure medical benefits are not cut. And finally, our research and data pillar, where 100% of our funds donated for research goes into the research grant programs. And since 2011, we have given a massive 5.1 million to different research projects. So to access our support and services and find more about MDFA and the work we do, please give us a call on our helpline and speak to our programs and services team, who are qualified vision scientists and optometrists. And you can also email the team and get the information, or you can go visit our websites to read more about MDFA and the work we do. And after this webinar, you'll have an opportunity to provide the feedback on the webinars and suggestions on hearing more webinars similar to this, one or other topics you'd like to hear more about. And there will be a link to past recorded webinars, and you can also register for other upcoming webinar sessions as well. So speaking of our upcoming webinars, our next webinar will be on the treatment landscape, the current and emerging therapies. It will be presented by Dr. James Wong. He is a medical retinal ophthalmologist. It will be on Friday, May 26th from 2 to 3 p.m. Australian Eastern Standard Time. It's tomorrow. So you can register through our website. You can scan the QR code on the screen with your phone or a smart device. Okay, Alice, now let's get some questions, audience questions.
Yep. So do you want me to read them? Yep. So The first question is on how far we are from stem cell therapy for the general public. Yep. So today there are some stem cell therapies for different conditions, not for AMD. So there's no, it's not available for the public when it comes to AMD. But we know this is not science fiction due to the clinical trials that are being made all around the world. And that goes to the next question, which is, do you think stem cell treatment would be expensive for me as a patient? If we are talking about AMD, because there's no therapy yet available with stem cells, you should not be paying for any therapy. If this therapy is to happen, it will be through clinical trial. And this is a very important point. Talk to your ophthalmologist, to your doctor, see if there are clinical trials that are happening. And no clinical trial should ask you for money. If you are asked for money for stem cell in the eye, there's a problem. Do not go there. Do not do it. This is not legit. So yeah, I hope I've answered those two questions together. Do you want me to go through the third question?
Oh, yeah, I will ask you the questions. Let me. So the one question is, I'm really interested in stem cell therapies. Where can I sign up to be the part of trials?
So again, I would talk to my ophthalmologist to make sure that you get the latest information, and ophthalmologists are the best person to talk to you as well. I'm assuming the MDFA would also be able to provide information as they receive it.
Yeah, and also I would like to add on this one. If you want to get registered yourself with different clinical trials and different organizations, then you can always give us a call. There are a few organizations like Center for Eye Research in Melbourne, Safe Sight Institute in Sydney. So if you give us a call on our toll-free number, we can provide you some links where you can register yourself for the clinical trials. So the next question is, how long does it take to find if a drug is suitable for the treatment?
It can take years, and unfortunately, that's a very long part of research, is to make sure that drugs that are found work, are not toxic for other events, and that they're effective in different systems. So having the model right is really important, because sometimes drugs work in a suboptimal model, and then years are lost because we found an effect on something that's not optimal. But from the moment you find a drug to the moment it goes to the clinic can take a very long time, depending on if the drug is a new drug or if it's a drug that's being repurposed. So there's a lot of effort right now in repurposing drugs that have already been approved for the condition, because that would really make the whole process quicker. So in short, not one type of answer. It can be very long, or we can perhaps find shortcuts with drugs that are already available for the treatments.
That's awesome. So that concludes today's webinar. So I would like to say a massive thank you to Alice for presenting today, and to all of you for joining this webinar. Thank you so much for everyone joining. Thank you, Alice.
Thank you, everyone.